Welcome to Bio class. Today we will discuss structure of protein, the bond linking monomers in a polymer, and the concept of metabolism. Okay, the first topic here, structure of protein. The protein here, look at here, protein, which is already we have discussed, heteropolymers containing the strings of amino acid. What is a heteropolymer? And if we consider another term is there the homopolymer. So let's know a little bit about it. Here this concept of homopolymer, it means that the same type of monomer, it is joined with each other. Okay, same type of monomer. Suppose this is the glucose. We saw earlier in case of starch, but in protein, here also the polymer, here also the chain of amino acid, but these chains here the amino acids are not always the same. It can be different in different. It can be valine, leucine, isoleucine, methionine, threonine, arginine. Okay, so 20 different type. So that is why it is called the heteropolymer. This polymer, but the different type of amino acids are there, which makes the protein. According to the structural level, and that the molecular structure, well, biologists describe the protein structure as a four levels. The labels are the primary, secondary, tertiary, quaternary. This four level it is discussed. In four level, that I think you can understand that according to complexity that it increased. It means the primary level, the simplest form, it is found. So let's see the structure, then we can able to understand better. Here it is, four level of structure. First one, you can see that primary structure, amino acid, that the primary structure level. So look at here the heteropolymer, you can see the different color, the bits like that, different colors are there. These are the amino acids, different amino acids formed. It joined in the peptide bond, and it is in straight forms. So it is this is called the primary structure. The first amino acid here it is called as N-terminal. Suppose it is the first, the N-terminal amino acid, and the last one amino acid is called C-terminal amino acid. Okay. So this one is a linear sequence. Here the peptide bond. Suppose insulin, ribonuclease is one of the example. Secondary structure, if we see that the secondary structure, it is a regular coil, spiral, alpha helix, random coil, parallel fold, or plate sets like this. So here the amino acids are there. I mean the sequence pattern is same, but here it is different. That alpha helical type of keratin, it is it is found in keratin and silk fibroin, all this it is there. Now tertiary structure, that irregular, the coil like structure, that it can be some folding or super folding, some complex twisting of a polypeptide chain. Uh, I mean, in the three-dimensional shape, it looks like that woolen ball-like structure, as it is described in your book. You can see. This tertiary structure, the myoglobin is one of the example that the tertiary structure. Quaternary structure, the aggregation of the two or more polypeptide. Here, the aggregation of the multiple polypeptides, which are as many as 4 to 6, to form the single functioning protein is called quaternary. Suppose these are the monomer. Hemoglobin, one of the example, it is the quaternary structure. Is very complicated structure. It is, but in these 4 form, the proteins are available. Just know that. Next part, nature of bond linking monomer in a polymer. Okay, so we already discussed the starch, protein, fat. These are the polymer, and it linked with that unit monomers like glucose, like amino acids, like fatty acid, glycerol. So here the bond. This part is already discussed, but then also once again I'm just reminding you that peptide glycosidic bond and some other bonds. All right. So here the first we can see this the in between the glucose and the glucose, that the glycosidic bonds. Look at here the water molecule comes out, this bond it's between alpha D glucose, and when in beta D glucose this link it is looks different. Already you can see in cellulose uh, this beta D glucose, lactose also it is there. Okay, so in peptide bond in between two amino acid, here amino acid 1, amino acid 2. Look at here that here R1, other you can get the one carboxyl group, one amino group. So here the amino group, and here the carboxyl group CWH, the amino NH2. Here the bond will appear and the water molecule it moves out. So peptide bond will appear here in between, so it converts into dipeptide. So in ester bond in between glycerol and the fatty acid, here is the glycerol, here is the fatty acid. There are 3, 1, 2 and 3. It can be similar or dissimilar. Here R, it can be similar or dissimilar. So when this triglyceride, that is why it is called triglyceride, that fatty acid number is 3, glycerol is 1. So and here it can form, you can see this, here the water molecule moves out, so that the bond will form. So this is the triglyceride or fat. Okay. So phosphodiester bond in nucleotides, you can see this part here in phosphodiester bond is formed between nucleotides, pentose sugar in the hydroxyl group and the phosphate group. You can look at here in here it is the H3PO4 phosphate, and here this is the pentose sugar. Okay, so the bond is formed here. This is called the phosphodiester bond. It is found in the DNA RNA. So this is also the very important. And the last but not the least, that double strand in here, which makes that DNA double stranded. That you know that here is one strand and here is another strands. The strands are anti-parallel, and it joined with the hydrogen bond. Between adenine and thymine there are 2 hydrogen bond. Between guanine and cytosine there are three hydrogen bond. So this hydrogen bond, um, it is joined with one another and form the DNA, our genetic material. So that also you have to keep in mind. Okay. Now, concept of metabolism. Concept of metabolism. Metabolism, it is the overall reaction in our body. So here it is discussed it is nicely that one of the greatest discoveries ever made was observation that all these biomolecules have a turnover. This means they are constantly being changed into some other biomolecules and also made from some other biomolecules. This breaking and the making is through chemical reaction constantly occurring in living organism. Together, all these chemical reactions are called metabolism. Metabolism is total reaction in our body. We can divide it into anabolism of and the catabolism, the nature of that reaction. But metabolism, it is a sum total of reaction, and here it is nicely described in your book, that turnover, that always that biochemicals it converts to some other biochemicals, and and again it converts into other biochemicals, and it forms and it utilized again. Again it can be formed new other way. Sometime it takes energy, sometime it gives energy. So energy is one of the side by side that point that you have to keep in mind. So in a very simple way that can be discussed in this, here it is discussed that in metabolic pathway, if we discuss the pathway, now there are two different pathway, anabolic and the catabolic pathway. Anabolic pathway, here the small molecules are assembled into large one. So they are separate with the help of energy. Energy is required. So these are joined together one complicated forms. We need catabolic. From this complicated forms, again it can separate, and the energy is produced here. This things is always going on in our body, and this total anabolic and the catabolic, the anabolism and the catabolism, we are collectively calling metabolism. Okay, so this is the concept, basic concept is this. So here catabolic and anabolic reactions are organized into series of reaction. It's called pathway, this metabolic pathway. Just now it is told that any inertial molecule, the compound or the substrate, it converts into several different forms, intermediate forms, and ultimately it's turns into the product. So A, if it is the initial molecule or the compound, then E will be the final molecule in the compound or product. Intermediate will be the B, C, and the D. Each step of the pathway is mediated by the specific enzymes. If we leave this A, B, C, and if you see one real, reactions that you can understand that here it is the glycolysis, which is there in your syllabus. See that the glucose, here the breakdown of glucose. So this is if you consider this is the initial product, the A, And after that, so many intermediate glucose 6 phosphate, fructose 6 phosphate, fructose 1 6 triphosphate, glycerol 3 phosphates. In this way, that it ultimately the final it comes the pyruvate or pyruvic acid, 2 molecules. So this is the structures you can see, and each and every in blue color you can see the each and every steps the enzyme is required. So enzyme mediated this reactions. So this is the one part of the metabolism, and the one example that you can consider. All right. So today up to here, the next day that metabolic basis of the living and the other part we can discuss.